Hi, my name is Shaheed.
My name is Joshua.
My name is Tseten, and our project is nutrient reduction and recovery in an algae-based wastewater treatment employing immobilized Volvox and Chlorella Vulgaris.
The aquaculture industry dumps 8 million tons of fertilizer into coastal ecosystems every year. As this number increases due to increasing industrialization, the mass extinction of these ecosystems may become a reality due to the fertilizer and other forms of runoff, creating eutrophicated environments. This promotes the proliferation of toxic algae that causes the region to be impossible to fish or drink from. In addition, these algae litter the top of the aquatic region, which prevents other aquatic plant life from receiving sunlight and thus completing photosynthesis. This results in highly deoxygenated water that makes it almost impossible for other organisms to survive within. These regions are known as dead zones, and there are already 400 of them around the world. It leaves only microorganisms and micronutrients to be left within the water, and the process of removing these substances, tertiary treatment, is the most inefficient and costly one of the three.
Once wastewater treatments were introduced, as shown in Figure 1, there was a significant decline in phosphate levels, one of the main nutrients causing eutrophication. According to previous research in testing the growth and nutrient removal in free and immobilized algae, as shown in Figure 3, the immobilized algae of S. obliquus showed a significant decline in protein and lipid content, and in Figure 2, displaying mean growth of the algae in artificial and regular wastewater. Basically, what this concludes is that immobilized algae can be utilized when effectively removing toxic nutrients. Our null hypothesis was that if algae cultures are placed in a eutrophic body of water, the nutrient content would not decrease in a 24-hour period, and it is not an effective treatment method. And our alternative hypothesis is that if algae cultures are placed into the eutrophicated body of water, nutrient and heavy metal concentration of the wastewater will decrease.
So overall, the data conveys that Volvox is superior in bioaccumulating far more copper and zinc molecules than Chlorella, the immobilized Chlorella. However, immobilized Chlorella was far more efficient at removing the other inorganic molecules, such as the nutrients. Volvox's efficiency can be explained through its much larger biomass than what Chlorella had accumulated, because it is a colonial growing type of algae. Therefore, it will obviously have more biomass than Chlorella vulgaris. And another factor that could have affected it, or an error that went wrong in the experimental process, is, according to past research, pH plays a huge factor in bioaccumulation by algae. It regularly affects trophic transfer between the cell membrane and the extracellular fluid.
As for future studies, this way of treatment can be used on a larger scale. This can be further utilized in the most optimal places in treating certain areas where eutrophication occurs most.
Thus, we plan to repurpose the algae as biofuel in order to ensure the process is as environmentally and cost-efficient as possible.
Hello, my name is Ellie Vaserman. My research project is entitled An Analysis of Microplastic Contaminants on Staten Island Shorelines. To begin, I would like to share my most shocking finding. Microplastics were found in 100% of the samples so far analyzed. Microplastics are pieces of plastic that are less than 5 millimeters in size. They are formed either through the degradation of larger plastics or as primary microplastics when they are directly manufactured in products such as hygienic products. Microplastics enter biotic ecosystems through ingestion and interfere with immune responses and increase toxicity levels in the body. In fact, a recent study estimated that the average human consumes a credit card worth of plastic every week only through microplastics. While microplastics are a ubiquitous and pervasive problem, very little research concerning their physical properties or how to remove them have been done. In New York, there have been no studies done on microplastics in shoreline sediments. Thus, the goals of this research were threefold: to create a methodology for isolating microplastics from sediment, to create a preliminary study of microplastics on Staten Island, and to record the properties of found particles in hopes of determining a potential source. Samples were collected on South Beach on the east coast of Staten Island. So far, 5 slightly varying methods were used. The most effective method so far was the fifth method. Here, the sample was divided into 3 size fractions: 1 to 5 millimeters, 300 microns to 1 millimeter, and less than 300 microns through dry sieving. The size fractions under 1 millimeter went through a hydrogen peroxide treatment in order to remove organic matter. They were then rested and settled in sodium chloride solution for 5 minutes, stirred for 2 minutes, and settled for 10 minutes. Suspended particles were removed onto a filter and observed under a microscope. Key properties of the microplastics, including shape, size, color, and appearance of erosion, were cataloged based on these images. As mentioned before, microplastics were found in every sample, for a total of 86 microplastics found across 9 samples. Method 5 has had the highest efficiency in terms of microplastics per unit mass, with about 0.2 microplastics per gram. In general, 69.8% of the plastics were fibers, and 57.9% of the plastics were small microplastics. The data implies that both of the hypotheses should be accepted. The hypothesis of a larger concentration of small microplastics was supported, since 66% of the microplastics found were under 1 millimeter in size. While some plastic material may have been lost, this count, as well as the recorded physical properties, could be an estimation to represent all of the samples. Furthermore, smaller sizes and fibers that were found indicate that these were primarily secondary microplastics. Since all of the samples contain fibers, there is clearly a larger source of these contaminants, which would likely be sewage or fishing twine due to similarities in appearance and color. In general, the high count of plastic fibers, as well as the presence of microplastics in all of the samples, indicate that further studies into microplastic contamination should be conducted, specifically focusing on how these particles have traveled. Thank you.
Hi, I'm Julian.
And I'm Sandhya, and before we talk about our project, we just want to contextualize why sustainability in the chemical industry is so important.
Starting out, the U.S. Energy Information Administration projects a 28% increase in world energy use by 2040, which is only 20 years away. And the chemical industry accounts for 26% of global energy demand. Additionally, the chemical industry accounts for over 5.5% of global CO2 emissions.
Now, electrochemical engineering is unique because it implements renewable energy sources to explore new chemical transformations, and it has the potential to amazingly impact over 75% of chemical products.
Nylon 6,6 is integral to automobiles and textiles and is considered one of the most useful polymers in industry. However, 70% of nylon production is currently thermal because the electrochemical route is unselective and isn't seen as a viable alternative for manufacturers.
Now, in an industry where cost and efficiency are the only vocabulary words, green chemical reactions just aren't that popular. But it really bothers us that we are constantly forced to choose between progress and sustainability. And at our lab, we've worked to make the electrochemical process to produce nylon significantly more efficient.
This process was first invented by Manuel Baser, who worked at the Monsanto company in the 60s, but due to its issues, it was not widely used. And before we got there, our lab was able to bring efficiencies up to 83% by testing specific factors in our electrolyte. But they left phosphate salts, which are largely responsible for charge transport, untouched.
And that's where we came in. We looked at 4 metal alkali cations in the phosphate salt and found that potassium phosphate actually works significantly better than sodium phosphate, which is what Baser used. And when we quantified how much better, we were shocked as we were able to improve the reaction to up to 97% efficiency. This outpaces efficiencies of both electrochemical and thermal reactions and could translate into huge improvements in nylon manufacturing and popularize the electrochemical production process.
And the good news is our system uses half the raw material and energy of thermal reactions, produces half the waste, and decreases production costs by about 30%.
And of course, we aren't going to stop here. These results can be applied to other polymers in the future with the goal of eventually transforming the chemical industry. Now, it might sound ambitious, but it only needs to be possible. Thank you.
Thank you.
Hello, my name is Eric Kim.
And my name is Kevin Gu. Our project is titled Highly Mesoporous Carbon Aerogel as Catalyst Support in Proton Exchange Membrane Fuel Cell. And this is the board for our project. We'll go through the details in each section slide by slide.
As reported by the EPA, over 29% of greenhouse gas emissions in the United States are caused by gasoline cars alone. To combat this issue, scientists have been exploring technologies like hydrogen cars that have long range and fast refueling time. Our project is about developing and using carbon aerogels in these hydrogen fuel cells and its application in mitigating climate change.
The hydrogen car is powered by a proton exchange membrane fuel cell, or PEMFC in short. It uses oxygen and hydrogen as fuel and outputs water through a redox reaction. And this reaction occurs on the electrodes, which consist of the catalyst and catalyst support. However, the low catalytic activity of these electrodes prevent this technology from being widely commercialized.
To improve the PEMFC performance, many scientists have focused on researching the catalyst material. We took a novel approach of improving the catalyst support, which is the foundation of catalyst performance. Our work is versatile because we can apply the improved catalyst support to any state-of-the-art catalyst to achieve even better results.
The issue with the current catalyst support, or carbon black, is its low mesopores, which are pores from the 2 to 100 nanometer range. Its high micropore count leads to low surface area, poor platinum distribution and utilization, which all contribute to poor fuel cell performance. Therefore, an effective catalyst support requires high mesopore volume.
So, we hypothesized that an optimized carbon aerogel with high mesopore volume will enhance fuel cell performance while allowing for an even distribution of platinum and increased platinum utilization. In our search for materials to use as catalyst support, we came across carbon aerogel. This material had been successfully applied to supercapacitors and was found to have a highly tunable pore distribution.
The first step of our work was to synthesize the carbon aerogel. We creatively modified a procedure from literature to best preserve the pore structure, decrease synthesis cost, and increase mesopore volume. The synthesis requires 4 steps: sol-gel polymerization, solvent exchange, ambient drying, and carbonization. We optimized the mesopore volume by modifying the RC, or resource element to catalyst ratio, in the sol-gel polymerization step, which has been shown to have the most impact on the mesopore volume. So, we synthesized carbon aerogels with RC ratios of 100 and 200 and 300 to observe the changes in mesopore volume.
We measured the pore volume and surface area using a gas absorption analyzer. From the pore size distribution graphs, we can immediately see that our optimized carbon aerogel has significantly higher mesopore volume than carbon black. And the data from Table 1 also confirms that high mesopore volume is correlated with high surface area. Ca200 has the highest surface area and mesopore volume, so we used this aerogel to continue with our experimentation.
After depositing the platinum on the catalyst support, we conducted transmission electron microscopy, or TM in short, to observe the platinum distribution. In the carbon black image, we can see a clear clumping of platinum, shown in the red circles, due to its low surface area. However, in our aerogel electrode, we observe a much more even distribution of platinum. This means that the platinum is much better utilized in the catalysis reaction in the fuel cell later on.
To quantify and compare the platinum utilization between the 2 electrodes, we measured the electrochemical surface area, or ECSA of the catalyst. We observed that the ECSA of aerogel is 2 times the ECSA of carbon black, which means that there is twice as much platinum being utilized in the carbon aerogel electrode.
After confirming that the high mesopore volume leads to high ECSA and even platinum distribution, we conducted 2 tests to observe fuel cell performance enhancements. We simulated fuel cell applications such as hydrogen cars and power plants by testing in atmospheric and oxygen conditions. We observed a 644% increase in maximum power density in atmospheric testing and a 705% increase in oxygen testing. This confirms that the high mesopore volume of the carbon aerogel leads to increased fuel cell performance.
And to ensure that our material is consistent in the real commercial use, we conducted a standard accelerated stress test, or AST. The durability is measured by the voltage loss at 0.1 amperes per centimeter squared after 10,000 AST cycles, which periodically cycles through the voltages to simulate 5,000 hours of use in a hydrogen car. We observed that carbon aerogel is 12 times more durable than carbon black, which means that carbon aerogel will increase the longevity of the electrode.
With our various characterizations and fuel cell testing, we observed that our hypothesis is correct. The high mesopore volume of carbon aerogel allows for an even distribution of platinum and increases platinum utilization, all of which to enhance the fuel cell performance. Additionally, we observed that our carbon aerogel can sustain its high performance in the fuel cell, much better than carbon black.
We expect that the high performance and durability of carbon aerogel will bring significant cost reductions in PEMFC technology. The 8-fold increase in power density after replacing carbon black with carbon aerogel theoretically indicates that we will bring down the electrode cost to about $23 per kilowatt of energy. This meets the 2020 Department of Energy goal.
We also observed that our carbon aerogel is 35% cheaper to manufacture than carbon black. This makes it commercially viable for hydrogen car manufacturers to switch over using our carbon aerogel.
Future work includes activating the carbon aerogel in carbon dioxide and further optimizing the RC ratio to increase the mesopore volume.
Thank you for listening to our presentation.
Good morning. My name is Jonathan Coulel de Beaufort, and I'll be presenting nanoparticles to power medical devices. Currently, pacemakers need to have their battery replaced every 5 to 10 years. This opens up the patient, literally, to risks of infection or even death. Therefore, this study presents a novel, cost-effective deposition technique of a material that can convert heat into electricity and is safe and flexible due to its unique organic-inorganic hybrid composition. The primary goal is to create membranes with thermoelectric properties. That is, membranes with the ability to generate electricity from a difference in temperature. The Seebeck coefficient reflects a material's efficiency in performing this conversion. As seen by the equation, the Seebeck is a number based on the amount of voltage produced for each temperature difference. So, since the membrane would then be inserted inside or on the body where there is a sufficient temperature gradient, it must satisfy rigorous constraints. It must be non-toxic, be cheap to manufacture, be efficient in its conversion from heat to electricity, and be flexible in order to be wearable. The material that best satisfies such constraints so far is tellurium P.PSS nanowires. Tellurium P.PSS has uniqueness of combining organic and inorganic materials with mediocre thermoelectric properties into an organic-inorganic hybrid with excellent thermoelectric properties. Tellurium is toxic since it is a heavy element. However, during the nanowire synthesis, the P.PSS polymer envelops the tellurium nanorod at a nanoscale, hence reducing the possibility of exposure to the environment. The product of this synthesis is a solid pellet. The advantage of this is by simply adding water, the particles become solution processable, which reduces manufacturing costs. Once in solution form, the particles are drop casted onto a flexible substrate and the electrical conductivity is tested. However, in real-world situations, the thermoelectric device would have to be resistant to mechanical stress. Thus, I designed an experiment to determine the effect of this stress. A half-pipe object was 3D printed and each film's conductivity was tested as the film was bent. The graph demonstrates a small hysteresis, but note that this stress of over 60 degrees will rarely occur in the human body. Now that the particles have demonstrated to be suitable for such applications, a method for depositing them had to be created. Current deposition techniques either do not work on flexible films or provide very limited conditions. Therefore, I built a 3D printer with an airbrush attached to it to deposit such particles on a substrate of up to 1 square foot. Synthesizing the nanoparticles and building the 3D printer were promising first steps. In the future, I'll measure the Seebeck effect as the mechanical stress increases. Thank you.
Hi, I'm William Hill.
I'm Amelia Ocampo.
And this is our project, Identifying Partisan Gerrymandering in Michigan. Every 10 years after the US Census, our congressional district lines are redrawn by each state legislature. However, each state legislature is often dominated by Republicans or Democrats, and they can redraw the district lines as they see fit, often to produce an advantage for their party. This is known as gerrymandering.
This past summer, in the Rucho v. Common Cause case, the Supreme Court ruled partisan gerrymandering constitutional. The court ruled that judging this matter is not within their reach because there is no limited and precise standard for evaluating partisan gerrymandering.
The goal of our project is to determine such a standard, one that could be applied everywhere to identify partisan gerrymandering. Our hypothesis was that Michigan was indeed gerrymandered due to the irregular shape of the districts, but this is only a surface level judgment, so it's far from conclusive. The first method we used was a computer simulation of the districts that used a Markov chain Monte Carlo method, which essentially simulates the distribution of votes across districts using the past data we have. And what we found with this was that many of the current districts were a far step from what a normal district would be considered by the simulation.
We also used geometric calculations such as compactness, which is something that each district is supposed to conform to by law. We used two different methods, the Polsby-Popper method and the Swartzberg method, both of which use the area and perimeter of the district to give it a compactness score from 0 to 1, with 1 being the most compact ideal district. We discovered that most of the districts had a score below 0.42, which is considered below average compactness.
We also used the efficiency gap, a method that aims to count the number of wasted votes in a given district. And so what we found with that was that around 1 million of the total Democratic vote in the state of Michigan was wasted in the 2016 election, and only around 740,000 of the Republican vote were wasted. And so that means out of every Democratic vote cast, only 50% of them counted in the election. And this compares to the Republicans, where only 40% of their votes were wasted.
Because our model found the districts made by a party are so unnaturally drawn to the point where they would rarely occur, it is very likely to be gerrymandering, therefore supporting our hypothesis.
Hopefully through a synthesis of multiple methods, with each indicating similar results, it is likely that gerrymandering has been committed. Hopefully this information can be used by the Michigan Redistricting Commission to make stronger, more informed revisions of the previous districts, one that avoid previous issues, and hopefully that can be used to create a more democratic America that prioritizes equal voter representation. Thank you.
My research focused on addressing some of the limitations of current genetic screening methods for pancreatic cancer. Pancreatic cancer is especially tough to diagnose early because symptoms only present themselves in the later stages, and current genetic screening methods have proven to be ineffective. These genetic screening methods are often based on genome-wide association studies, or GWAS, and I hypothesized 2 reasons why these studies may be ineffective at early diagnosis. First, genome-wide association studies are purely based on association. They don't tell us anything about what the genes that they're detecting are actually doing to contribute to pancreatic cancer pathogenesis, and therefore those genes or SNPs may just be peripherally involved in regulatory networks and won't help us with early diagnosis, catching the driver mutations. Second, the majority of these studies have been performed on the Caucasian population. This is especially detrimental for pancreatic cancer because incidence is up to 70% higher in the African American population than in any other racial population. I thought I could combat these 2 problems by looking at chromatin accessibility. The way I did that is I analyzed attack sequencing data, which is basically genetic sequencing of chromatin accessibility, and I analyzed that for 5 different pancreatic organoids that mirrored the different stages of pancreatic cancer using the R coding language. I found 2 main things. First, that chromatin accessibility does play a significant role in pancreatic cancer pathogenesis. The accessibility landscape varied greatly between the different stages, and this is important for understanding why incidence might be higher in a racial population, because chromatin accessibility is the main point of variation between racial populations. Many studies have shown that chromatin accessibility is the main source of variation. With further investigation, we can look at that to understand why incidence is so much higher in the African American population. The second thing that I found is that the SNPs or mutations found in GWAS were all on the periphery of highly accessible regions. Why is this important? That means that they're modulating accessibility to the actual driver genes of pancreatic cancer. This is exciting because if we can isolate the driver genes that are producing cellular mechanisms that are actively contributing to pancreatic cancer pathogenesis, we can target them with treatment. Before, we just had these SNPs that were modifying regulation, so we could not target the cellular mechanisms that they were producing. Another important application of this project would be to create a more equitable and effective genetic screening method that combines both GWAS data and chromatin accessibility data. That would address the issue of not knowing whether these genes or SNPs that are being detected are in accessible regions that are therefore being actively transcribed. And it would give us clues as to why the African American population has so much higher of an incidence of pancreatic cancer because, like I mentioned, the chromatin accessibility landscape varies greatly between racial populations.
I will be presenting my research about how I created the first objective quantitative method for creating adequate training datasets for artificial intelligence to run diagnosis based on pathological images. Artificial intelligence is everywhere in our daily lives. It can also be applied to the cancer pathology field, where it can improve the accuracy of diagnosis and help expedite treatments. But first of all, we need to develop large standardized image libraries to train these algorithms. Because of conditions in the lab, such as suboptimal lighting conditions or staining inconsistencies in producing hand-stained tissue specimens, standardized image libraries need to be developed to ensure the accuracy of artificial intelligence and machine learning programs. Standardization techniques, also known as color normalization, are one potential way to prepare images for standardized image libraries. This is the standard method that experts use to correct adverse image conditions, and it's currently done on an image-by-image basis. Over time, multiple methods of normalization have been developed, leaving pathologists with many techniques to choose from.
However, there are still a few obstacles in the way of producing effective images for image libraries, which experts have been unable to resolve. The first problem being, which method of standardization works the best, given the type of tissue or image imperfection? The second problem is, how can we establish that the normalized image is good enough for machine learning datasets? The answers to these questions lie in my research, which creates a validation framework to establish an objective way of comparing the results of normalization methods. The framework basically involves a couple of key steps, the first of which is intentionally changing an image to simulate a potential laboratory condition. By using a computer-based method to alter the images, I have an image with an adverse image condition in order to normalize and the original image. The second step is to perform color normalization. And finally, the third, most important step is to compare the normalized image to the original image using my new formula. Using this quantitative approach and working with pathology tissues allows pathologists to use the validation framework over and over again to make use of trial and error approaches to uncover more about cancer pathology and normalization techniques. This will eventually allow experts to expand image datasets and can pave the road for artificial intelligence in the pathology field. In summary, my research allows pathologists to use a mathematically objective method to quantify the effectiveness of different computer-based methods of preparing cancer tissue images, which therefore allows pathologists to utilize artificial intelligence approaches in dealing with cancer pathology images and diagnosis. Thank you, and enjoy the rest of the presentation.
Hi everyone, my name is Sam Singer, and today I'm going to be talking about disparities in the prognosis of cancer that are correlated with race and ethnicity. While researchers use a large number of genetic databases to drive our abilities to diagnose, treat, and prevent cancer, the leading cancer genome database, the Cancer Genome Atlas, has been shown to underrepresent minority races and ethnicities to a statistically significant degree. Other studies have shown that African American and Hispanic patients are more likely to experience poor cancer prognoses, as shown by higher tumor stages, higher death rates, and less treatable tumors, even after adjusting for the quality of medical care. As a result of this underrepresentation on genetic databases, the unique cancer profiles of these minorities that contribute to their differences in cancer outcomes are not well understood. We hypothesize that a high degree of genetic variation in tumors, called intratumor heterogeneity, is the cause for worse cancer outcomes in African American and Hispanic patients. To test that hypothesis, we sequenced the genomes of 62 patients, selected using a power analysis so that statistically significant data could be obtained. No globally statistically significant differences existed in mutation frequency between groups of different ethnicity. Next, we performed a gene-by-gene analysis of these mutations. For each gene, we filtered the mutations returned by genetic sequences to be left only with pathogenic or cancer-related mutations. Then, we checked each pathogenic mutation either for a significant difference in mutation frequency between races and ethnicities, or for a high raw mutation frequency in a minority group. Finally, we filtered for statistical correlation with the defined criteria for prognosis, those being tumor stage, death rate, and recurrence rate. In this manner, we obtained the pathogenic mutations that were correlated with poor prognosis in African American and Hispanic patients. No significant confounding factors were present, which allowed us to determine that there were 8 specific genetic mutations correlated with poor prognosis in African American and Hispanic patients. This conclusion, in turn, allowed us to infer that specific genetic mutations, not overall mutation frequency, are correlated with poor cancer prognosis in minority races and ethnicities. In the future, we hope to validate these mutations using a larger sample size, as well as use animal modeling to confirm a causative relationship between these mutations and cancer prognosis. Understanding of mutations that influence cancer prognosis can result in a better understanding of the cancer profiles of minorities and the development of target molecules that counter these prognosis worsening factors.
My name is Andrew Sosnov, and my research is on Alzheimer's disease. Even though I am very young, and I am not at immediate risk of this disease, there are many other people who are. In the US, there are over 5 million elderly people that are diagnosed with this disease every year. They suffer from memory loss, cognitive decline, and brain shrinking due to neuronal death. This disease has been first described 100 years ago, Yet, we still have no cure and no clear disease mechanisms for Alzheimer's. I examined a novel pathway for Alzheimer's disease involving the damage done on the mitochondria. Recently, it has been shown that presenilins, or coenzymes that participate in the cleavage of the amyloid precursor protein, or APP, to create the toxic product beta amyloid, has been found in the regions where the mitochondria interacts with the endoplasmic reticulum, also known as the mitochondria-associated membrane, or MAM for short. We tested mouse fibroblasts with presenilin deletion and human fibroblasts from samples collected from the patients of Alzheimer's disease and compared them with control subjects. So what did we find? Well, in both the mouse and human fibroblasts, we found increased lipid accumulation in the fibroblasts that are near the nuclei, suggesting that they may be in the regions of the MAMs. In addition, the accumulation of lipids are greater than those of the control subject. So what does this mean? Well, the inhibition of presenilins gives rise to the production of another product of APP, C99. C99 is responsible for binding with the aberrant lipids and is toxic to the mitochondria and will lead to cell death when these lipids are metabolized. This research can prove to be very useful for explaining and helping to explain the full molecular pathway involved in Alzheimer's disease and even possibly help with treatments for Alzheimer's disease in the future. I hope one day, when I become of age for Alzheimer's disease, that I won't have to worry about it anymore, as we will have a cure. Thank you for listening.
Hello, I'm Seamus McNulty, and I created a virtual self-administration laboratory with extended capabilities. What does that mean? Well, self-administration is when a human or other animal doses itself with a drug or substance. In this case, it is cocaine. When trained, a rat will reliably self-administer cocaine according to the compulsion zone theory of self-administration. The model holds that a rat will reliably self-administer cocaine when trained below a certain concentration known as the satiety threshold. Above the threshold, the rat will cease to self-administer. This model is vital for our understanding of addiction. However, the in vivo testing of rats requires a significant amount of funding and animal handling experience, limiting the amount of testing and teaching that can be done in the field. Therefore, there is a demand for a simulation that can capture the patterns of self-administration without the cost or training usually required, and one that is capable of being used as a tool of instruction for those unfamiliar with the field. Therefore, I created a virtual self-administration laboratory with extended capabilities. The simulation accurately captures self-administration sessions, as the intervals of self-administration between the simulation and 7 real-life sessions was found to have no significant difference, according to an ANOVA test. The simulation was constructed in C# and allows for the control of several variables and aspects of real-life simulation sessions. The speed of the session, along with the dose and the satiety threshold, can be increased and lowered. Additionally, the simulation allows for the injection of a competitive substance or antagonist, which allows for not only the interaction between rats and cocaine, but cocaine and various other substances. This feature of the simulation is in its alpha stages, but is planned for expansion. In the upcoming months, I will be working on improving the simulation to include more complex features of self-administration. As of now, and in the future, this simulation will allow for self-administration to be observed and taught, furthering research into the field of addiction and allowing for progress to be made towards treatment.
Hi, my name is Neha, and my project is identifying bacterial swarming idiosyncrasies via microgear rotation and confinement. So through last year's and this year's research, I developed a novel visual-based quantitative methodology for distinguishing between bacterial swarming and swimming, which has numerous applications, particularly in the fields of disease diagnosis and bioengineering. Bacterial swarming is qualitatively defined as a coordinated two-dimensional movement across a semi-solid surface, and bacterial swimming is defined as an individualized three-dimensional movement in liquid medium. Aside from my research, there are no quantitative ways to distinguish between these two motility types, and relying on qualitative distinctions can be highly unreliable and subjective. The reasons why bacterial motility is super important and having mathematical distinctions is important are as follows. 1. Bacterial motility is an indicator of host pathophysiology, which means that swarming is an indicator of disease or microbial imbalance, and swimming is an indicator of physiological normality. 2. Antibiotic resistance is associated with genes regulating swarming in many bacterial species, so having concrete distinctions in motility is crucial. The goals for my experiment were as follows. 1, to uncover distinctions between swarming and swimming motilities in a system of my design, and 2, to optimize my technology for applications in bioengineering and medicine. I used photolithography to create PDMS confinement sheets and microgears to which I added swarming bacteria and swimming bacteria and noted their biophysical properties, namely torque and power. And what I noticed was swarming bacteria had a higher torque on average than swimming cells. Then I zoomed in using particle image velocimetry to find difference in patterns of the different motility types by plotting the vectoral motion, and then I used computational simulation, namely the VICSEC model, to uncover the mathematical variables that was driving these pattern differences in swarming and swimming bacteria, and I found that the intrinsic noise variable was the one that was responsible. A noise variable indicates a particle moving contrary to the group, and the intrinsic part of that variable means that the environment is not influencing its motion, but rather there is some internal factor causing it to diverge, and I found that the biological factor that could be contributing to this mathematical variable is cell length, because swarming cells are typically longer than swimming cells. The technology that I used and designed could be applied to diagnosing disease because it shows clear visual and quantitative distinctions between swarming and swimming to inform diagnosis and appropriate treatment. Usually it takes several days to get the results of a urine culture for diagnosing a UTI or for a fecal culture for diagnosing gastrointestinal disease. My system takes just a few hours, and it can successfully determine the presence of motile bacteria and inform early appropriate treatment. It can also be applied in bioengineering because the technology can be used in microfluidic circuits without the need of power, since the bacteria would be able to generate sufficient torque and power in substantial numbers to sustain the circuit biologically. Thank you for listening.
Hi, I'm Sophie Marcus Wade, and I work at the Deitch Lab at Weill Cornell Medical College under Dr. Kirk Deitch and graduate student Victoria Schneider. The goal of my lab's research is to better understand malaria parasite Plasmodium falciparum's way of evading the immune system in chronic infections, with the ultimate, much more lofty goal of helping to create a more effective and cheaper treatment for the type of malaria that Plasmodium falciparum causes. So Plasmodium falciparum has a life cycle in the human red blood cell, and it is during this life cycle that it places a protein called PFEMP1 on the outside of an infected red blood cell. This PFEMP1 is how the immune system identifies the Plasmodium falciparum as a threat. But each parasite has about 60 different variants of PFEMP1 that all appear differently to the immune system. So Plasmodium falciparum can effectively change the way it appears to the immune system by switching the variant of PFEMP1 that is on the outside of an infected red blood cell, making the infection caused by the parasite much harder to fight. Each PFEMP1 variant is encoded by a VAR gene, so there are about 60 different VAR genes, and only one VAR gene can be expressed at a time, which means that only one variant of PFEMP1 can be on the outside of an infected red blood cell at any given point. This mutually exclusive gene expression is controlled epigenetically, so the DNA with the VAR genes wraps around a collection of 8 histone proteins called a histone octamer, and DNA that is wrapped tightly around the histone octamer cannot be used to create mRNA, and DNA that is wrapped loosely around the histone octamer can be. Which DNA is wrapped tightly versus which is wrapped loosely is determined by the placement of methyl groups on these tails that come off of histone proteins. Many of these methyl groups come from an organic compound called SAM. Plasmodium falciparum gets its SAM by taking methionine from the host and using an enzyme called SAMs. So in order to test one aspect of the relationship between SAM levels and VAR gene switching, I measured the VAR gene expression of parasites with normal SAMs levels and knocked down SAMs levels by measuring the mRNA of all 60 different VAR genes that the parasites I worked with had. If you look at my poster, you can see over 3 weeks, the VAR gene expression in the parasites with normal SAMs levels and the parasites with knocked down SAMs levels remained the same, with the same VAR genes being expressed in about the same proportions. What this means is that a SAMs knockdown does not increase VAR gene switching, which makes sense because less SAMs results in less SAM, which would lead to fewer methyl groups to methylate histones and increase or induce a VAR gene switch. Thank you.
Hi everyone, I'm Rafik.
I'm Tangina.
I'm Raisa.
So in your high school chemistry class, you probably learned about nitrites and nitrates and compounds containing them. Well, in the meat and dairy industry, they're used as extremely effective chemical food preservative. But there's an issue with their use. When nitrites and nitrates come in contact with the amine functional group, which is nearly ubiquitous in meat and dairy products, nitrosamines are formed. Now, these nitrosamines have been shown to cause gastric, bladder, nasopharyngeal, and esophageal cancer. Nonetheless, they are still used despite the potential health hazard, with the FDA actually regulating the dosage instead of banning the use altogether. This inspired us to try and find an alternative that could be just as effective and harmless in consumption.
So we actually went through a number of studies and a lot of things stood out to us. Organic acid producing bacteria prevented the growth of P. chrysogenum, and an acid mixture enhanced bacterial inhibition in the preservation of raw pork more effectively than individual acids alone. These studies inspired us to combine ascorbic acid with sodium citrate in an effort to reduce the growth of fungus on cheese that we cooked and plated ourselves in the lab. The fungus we used was Aspergillus niger, which is a plant-infecting fungi from the Aspergillus family. Consumption of foods that are contaminated with A. niger may lead to allergies and stomach pain or cause even more severe effects, such as the development of otomycosis or fungal ear and pulmonary infections.
We created our own cheese and used a combined mixture of ascorbic acid and sodium citrate, which has never been done before, to act as an alternative to nitrates, and then we measured the fungal growth on our cheese. Overall, we found that the higher concentrations of ascorbic acid was most preventative of the fungal growth because the cheese we used with the higher concentrations had the least Aspergillus niger growth. In the future, we would be interested in testing our solutions on different products that typically are preserved using nitrates and nitrites. Ultimately, we enjoyed stepping up to address a problem in our food industry.
Thank you for listening.
Take care.
Stay safe.
Hi, my name is Thomas Larson, and this is my research project. Cloud chambers are devices capable of showing the movement of alpha radiation, otherwise invisible to the naked eye. Despite being very useful, they are often criticized for their inconvenient size and have therefore been abandoned. Introducing the microcloud chamber, which uses background radiation to replicate the effect of a full-size cloud chamber on a miniature scale. The cloud chamber is split into two halves, which work together to create the necessary conditions for cloud formation. It also uses digital microscopes to capture and broadcast a video of the experiment in real time. This video output is fed through a server which I created, which can be accessed by anyone, anywhere on the planet. My research project was to examine if my microcloud chamber could provide a means to demonstrate and broadcast video of the presence of alpha particles with a specificity and or sensitivity of at least 75%. My experiment consisted of the monitoring of the supersaturated vapor inside my cloud chamber and the observation for any instances of ionizing radiation tracks using the digital microscopes. This was performed about 10 times, with each trial lasting around 15 minutes. The specificity of this chamber was concluded to be 75%, while the sensitivity of the chamber was only 67%, as seen in Table 3. This chamber was able to detect instances of alpha and beta radiation, as seen in Figure 8. As far as the telemicroscopy, the experimentation was successful in showing that a user anywhere on the planet with access to the IP address and password can remotely connect to the video feed of the server in real time. While the specificity of the chamber met the 75% requirement as set by the research question, the sensitivity fell slightly short. This was likely due to the height of the chamber. With more adjustment, the sensitive area in the chamber can be expanded, leading to a higher overall sensitivity result. Potential applications of this technology include being an accessible tool for students in physics classes to further their understanding of the atom, and as a solution to the limited market for alpha radiation detectors. In conclusion, this chamber was successful in demonstrating the functionality of a full-size cloud chamber on it while still being compact and inexpensive. The microcrystalline ice tracks have varying shapes and velocities, indicative of alpha, beta, and gamma radiation from background sources. In the future, my microcloud chamber will be tested using Rutherford's famous gold foil experiment, using zinc sulfide and video micrographs to capture changes in the alpha particle trajectories as they strike the gold foil. This concludes my presentation on the microcloud chamber. Thank you for your time.
Hello, my name is Jeffrey Munsell, and I'm a senior at the Bronx High School of Science, and I'm talking to you today about candidate selection of Aurora. And the interesting thing about Aurora is that they're a type of star, and they're a type of star which is special because they're variable, which means that over time the brightness of the object varies as they're expanding and contracting. And most objects in the night sky are going to be non-variable. So this is a good characteristic that we can use to identify what potentially might be our Lyrae from everything else out there. And we want to find these objects because they're what are called standard candles. And a standard candle is an object that you can accurately determine the distance from us to it. So what that means is that when we find these Aurora Lyrae, then we can get these anchor points, these known points out in the Milky Way. And if we find a bunch of them, then we can start to use them to investigate the structure of the Milky Way and see how it looks out on the outskirts that we still don't know much about, and we don't know how far it extends out. So that's the idea, and the way that I'm doing it is through a survey called the Next Generation Virgo Cluster Survey. And this is a survey that was done to focus mainly on distant objects, on the galaxies in the Virgo cluster. And while there's other galaxies that are in the images, what I'm looking at is actually the foreground. I'm looking at, in the Milky Way, these Aurora Lyrae, which are also in the data set. And this data set, what it has is, over years of data, there are multiple measurements for each object. So I can look at these different objects, the objects over time. So I can see this object was measured at these different points in time over this time period of months or years, and I can see how is it varying over that time.
Hey, I'm Z from San Jose High School, and I study Balmer studies absorption lines and pulsar-burst with E+A galaxies. Through my in-depth analysis, which we'll get into later, my data showed an idea opposing what was traditionally accepted regarding E+A formation is correct. But first, what are E+A galaxies? Well, these are galaxies that have been through the starburst phase, a phase where a galaxy is undergoing a tremendous amount of star formation, hence the alternative name post-starburst. E+As are specific galaxies known for these characteristics. Looking at the spectrum graph, which shows luminosity at each wavelength, we can see a small upward spike at the beginning and an overall downward slope. Furthermore, we can see dips in the Balmer series absorption line, hydrogen alpha, beta, gamma, and delta. These dips or absorption lines have an equivalent width, which indicate how strong the absorption line is. These are all characteristics of E+A galaxies. But why is all of this important? Well, the E+A phase is a significant part of galaxy evolution, since researchers have estimated approximately 30% of the elliptical galaxies go through the E+A phase at some point in their evolution. Each galaxy also has a morphology, the shapes or appearances. Galaxy morphologies are important because they give a hint at how galaxies are formed. Through this research, I wanted to evaluate whether there's a correlation between the morphologies of E+As and their Balmer series absorption lines. Previous research suggested that mergers in galaxy-galaxy interactions, which relate to violent events, could be the mechanism triggering the E+A phase. Since they only discussed these concepts briefly, I wanted to go in depth with my research on this relationship specifically, and I hypothesized that there is a positive correlation between the galaxies' equivalent widths and morphologies, meaning that the formation of E+A galaxies depend on their morphological or dynamic differences. In my study, I used various forms of data collection: the Sloan Digital Sky Survey for galaxy sample selection and spectrum files, a Python computer program IRAV for PyRAV for computing the equivalent widths for E+As, and the NASA Sloan Atlas for obtaining the SIR6 indices of the selected galaxies, and this provides information on their morphologies. To analyze for a trend between the collected data, several SIR6 index versus equivalent width graphs were created. This result is kind of puzzling at first glance, so I made a flowchart to guide us through. Absorption lines are caused by A-type stars in E+A, which absorb light. Thus, large amount of A-type stars in a galaxy indicate an E+A. Meanwhile, more violent events in a galaxy's lifetime mean there is an elliptical morphology, while undisturbed galaxies tend to have a spiral morphology. And the SIR6 index tells us the specific morphology each galaxy has, higher meaning more elliptical and lower meaning more spiral. In my data, I found varying absorption trends and varying SIR6 indices, but was there a correlation between equivalent widths and SIR6 indices? No. This can be seen through the near horizontal lines in the graph and the close to zero R-squared values. Thus, the amount of A-type stars are not dependent on the galaxy's morphology. Ultimately, this means that events or the absence of events a galaxy experiences in its lifetime does not affect whether it develops into an E plus A galaxy. Overall, these results oppose the proposed hypothesis that there was a correlation. To explain this, while some studies touch on the idea that disruptive events could lead to the E plus A phase, the research only involved 21 E plus A galaxies, and only 5 out of the 21 have violin-related morphological features, in comparison to the 50 galaxies and 200 absorption lines I analyzed.
Hi, so my name is Nyekachima Agwu, and I'm from the Bronx High School of Science. The title of my research project is Computational Modeling of Bioactivity and Botanical Kombucha Analogs, How to Survive the Post-Antibiotic Era Using Fick-Second Law of Diffusion. So, what is the problem? The problem is that bacteria have been developing resistance to pharmaceutical antibiotics for years, and this not only poses problems for people in third world countries who can't afford to keep buying new antibiotic treatments, especially people with diseases like HIV that require several kinds of antibiotics. It's also not as safe as we think, and it poses this looming era, the post-antibiotic era. So what I decided to do was use botanical herbs, calendula, meadowsweet, and ginkgo in combination with kombucha to test its effectiveness against Citrobacter frundi and E. coli. So I used the disc diffusion assay test to carry out this, and it's very simple. You can even do it in the kitchen. And then I used Fick's second law of diffusion to model it so that it shows basically that a more effective antibiotic will be the one that has, as time goes on, more absorption in the agar and more area covered because it's able to kill more of the bacteria. So my results were that meadowsweet was more statistically significant than ginkgo calendula and just kombucha alone. So it shows that it not only increased the effectiveness of kombucha against bacteria, but it also is comparable to the positive control, which is chloramphenicol. So what does this say in the big scheme of things? This says that while meadowsweet and other botanical herbs cannot be used yet as a substitute for pharmaceutical antibiotics, however, it can be used as a supplement and an adjuvant with these pharmaceutical antibiotics because meadowsweet in particular and other botanical herbs have different phytochemicals in them that because they've grown up so long with these bacterias, they've developed their own defense mechanisms against them. So it makes it hard for these bacteria to develop resistance to these botanical herbs. So in the big scheme of things, this is safer and it's more affordable for people around the world. So it can possibly delay the post-antibiotic era. So that is an overview of my project, and I hope you liked it. If you need any more clarification, you can shoot me a question in the discussion box. Thank you.
Imagine you're a farmer and you just added fertilizer to all of your crops. The following day, it rained and everything washed away. What do you do? Hi, my name is Yushan.
Hi, I'm Shaheed.
I'm Ethan. And our project is about the global fungal network, which is made up of mycorrhizal fungus that forms a symbiotic relationship with plants within the network. The hyphae of these fungus serves to facilitate plant communication and the transfer of essential nutrients such as potassium. We have read about this form of network being in nearly all ecosystems naturally. We wanted to see whether such a network can be recreated artificially in a laboratory setting, as well as the effects of the additional fungus. This is because of what we have read about the concerning practices of agriculture.
Farmers often add an excessive amount of commercial fertilizer to meet high food demands. The abundance of fertilizer remaining in the soil leads to runoff into bodies of water, causing algae blooms that blocks up sunlight. This in turn decreases the oxygen level for sea animals, disturbing the ecosystem. We want to explore the possibility of reducing fertilizer use through adding additional fungus into the soil. So our hypothesis is fertilizer nutrients added to one side of the planter box containing soil might be transferred to the low nutrient soil region by the addition of the fungus Aspergillus, thus behaving as a fungal network.
So now let's look at the results. In the first graph, through comparison of the potassium level of the side that we didn't add any fertilizer to the side that we added fertilizer and additional fungus aspergillus, we can see that there's a more obvious increase of the potassium content on the non-fertilizer added side. This suggests that the additional fungus in the soil helped nutrient transport. Looking at the second graph, with a comparison of the potassium level of all the non-fertilizer added side in our experiment setups, we can see that all of them have an increasing trend, which suggests that fungus were aiding the nutrient transport. In the experimental group, even after the beans have consumed most of the nutrients, the leftover potassium transport by the additional fungus was still a lot compared to the control group. This graph overall demonstrates how effective the underground soil fungal network is in transporting nutrients. Lastly, let's shift our focus to the third graph, which is comparing the fertilizer added sites in our experiment setup. The result is pretty self-explanatory. Because we're adding fertilizer daily into this site, the increase in potassium level here is no surprise to us. In conclusion, all of these have suggested our hypothesis that additional fungus in the soil will indeed help nutrient transport underground. At the end, we would like to thank Dr. Paolo, Dr. Margarita, and Dr. Matthew for providing us with the laboratory settings and all the material we need for completing this project. Thank you for listening to our presentation.
Hello, my name is Rohit Matthewshin, and today I'm here to talk about my experiment, Won't You Be My Neighbor? Every 5 seconds, 20 babies are born. By the year 2100, there will be an expected population of 12.3 billion people. Now, the planet has a limited amount of living space and cannot harbor this population boom. This begs the question, what can we do from here? Now, a new location for us to live in must reside in the Goldilocks zone, which, just like the story, must not be too hot, not too cold, just right. So, in our solar system, one of the perfect spots is Mars. So Mars is covered by sand, dirt, and dust. It's very iron-rich. It's called regolith. Now this here is a simulant, a Mojave Mars simulant. It's quite hard to see right now because of the bag, but that's how it looks. So Mojave Mars simulant, or Mars soil itself, the regolith, is actually very nitrogen deficient. So I tried fixing this by using soybeans. Now, soybeans are very, very rich in proteins, carbs, and lipids. They are very, very important because they can grow in many different locations. They're easy to grow, and they are very good for places that have growing populations. Now, soybeans have a property called nitrogen fixation. Now, we can use this by using companion farming, which is where you pair one plant with another. It's because it'll help them grow. Now, in this experiment, we paired them with millet, which is very hardy and can grow in dry regions and has a very quick maturing rate. So in this experiment, we compared rockwool or hydroponics to compost as well as the Mojave Mars simulant. You guys can see the method. And now moving on, we'll talk about the results. If you look at table 2 first, you can see that all of the Mars groups start a deficient amount of nitrogen. But at the end, every group that had the soybeans in them all became sufficient. Now, what this means is that over time, they were able to pump nitrogen into the regolith and actually help the plants grow. The 2 groups that had no soybeans in them at all experienced no change in the amounts. Now, you can also see throughout all the groups, as there were more soybeans and more soybeans per group, there's actually higher biomass, higher dry mass, as well as the mean millet height actually grew with more soybeans. Now I'll talk about the discussion. Now we want to tie our plants so that they can grow straight, and we as well want to make sure that our plants all stay pinned down. Now our hypothesis was supported because the MMS soil became more nutrient rich with nitrogen-fixing plants, and the amount of legume plants increased, as well as the biomass and the dry mass. Thank you.